Assalamualaikum. My name is Muhammad Ali Haider, and today I will be delivering lecture on pharmacology. This will be lecture number 9. Permeation. Drug permeation proceeds by several mechanisms. Passive diffusion is an aqueous or lipid medium is common, but active processes play a role in the movement of many drugs, especially those molecules are too large to diffuse readily. Aqueous diffusion. Aqueous diffusion occurs within the large aqueous compartments of the body, interstitial space, cytosol sector, and across epithelial membrane, right, tight junctions, and the endothelial lining of blood vessels through aqueous pores that in some tissues permit the passage of molecules as large as molecule weight of 20,000 to 30,000. Diffusion. Aqueous diffusion of drug molecules is usually driven by the concentration of gradient of the permeation drug or a downhill movement described by a fixed law. These blows drug molecules that are bound to large plasma proteins, for example, albumin, do not permeate most vascular aqueous pores. If the drug is charged, its flux is also influenced by electrical fields, for example, the membrane potential, and in parts of the nephron, the transtubular potential. Lipid diffusion. Lipid diffusion is the most important limiting factor for drug permeation because of the large number of lipid barriers that separates the compartments of the body. Because these lipid barriers separate aqueous compartments, lipid aqueous partition coefficient of a drug determines how readily the molecule moves between aqueous and the lipid media. In the cases of weak acids and weak bases which gain or lose electrical charge and bearing protons depending on the pH, the ability to move from aqueous to lipid or vice versa varies within the pH of the medium. Because starch molecules attract water molecules, the ratio of the lipid soluble form to lipid soluble form for a weak acid or weak base is expressed by the Henderson-Hesselbus equation. Special carrier molecules exist for many substances that are important for cell function and too large or too insoluble in lipid to diffuse passively through the membrane. For example, peptides, amino acids, and the glucose. These carriers bring about movement by active transport or facilitated diffusion, and unlike passive diffusion, are selective saturates and inhibitors. Special carriers. Many cells also contain less selective membrane carriers that are specialized for expelling foreign molecules. One large family of such transporters bind adenosine triphosphate and is called ABC ATP binding cassette family. This family includes the P glycoprotein or multi-drug resistance type MDR1 transporter found in the brain, testes, and other tissues, and in some drug-resistant neoplastic cells. Similar transport molecules from the ABC family, the multi-drug resistance associated protein MRP transporters, play important roles in the excretion of their metabolites into urine and the bile, and the resistance of some tumors to therapeutic drugs. Special carriers. Several other transporter families have been identified to not bind ATP but use OIN gradient to drive transport. Some of these the solutes carriers SLC family are particularly important in the uptake of neurotransmitters across nerve ending membranes. Thank you very much. Inshallah, I will be giving more lectures on pharmacology. Stay tuned and subscribe to my channel. Take care. Allah Hafiz.